share right now. Just give me a few seconds. I'm trying to solve. Okay,
Okay, we are live in Facebook.
Just a second, guys. I'm trying to solve the sharing the screen from this computer. Okay, not easy because I am on different computer than last time. So sorry for it. No, please be patient. Be patient. I'm sure I will manage. What's here? Screen. Share screen. How are you all? Sorry for waiting, but
no, don't worry. So we usually in academic culture we live.
Okay, so that's great. Good to know. So I hope I will not use much of your time. Share screen. Much better. Okay.
Okay, perfect.
Oh, perfect. Do you see it?
Yes.
Yeah. Super.
So we are ready. Welcome again, David, and it was a pleasure for me and also for students to have you again here as guest. And we were so impressed from your research work. But I think that today we will talk about some very exciting topic on great apes and primates and possible transmission of parasitic and other kind of zoonosis. So I don't want to take you so much time. Welcome and good lesson to everyone.
Thank you very much for a nice introduction. I hope everything will work well. So it's my pleasure to speak today about parasites of African great apes, which is one of the really main themes my team is working on. And I'll try to combine in this lecture a little bit of nice pictures of Africa with some heavy science based on the genomics of the parasites and next generation sequencing and so on. But in many places, I'll not really go into detail, not to bother you with technical details. And anyway, great part of these results are results principally obtained by my co-workers, PhD students, and postdocs in our team. So I really cannot say that I am the author of all the data I'm gonna to present here. So this is one of the most photographed gorillas. It's Macumba, the leader or silverback of group of lowland gorillas in Central African Republic where we work. And it's one of the really most photographed lowland gorillas in the world because it's one of the oldest habituated groups. I'll go to that later. So why to speak about the parasites of great apes? Why to bother about something that remote and exotic and so on as the great apes are and their parasites somewhere in the forests in Africa? I think there is a range of good reasons. The first reason is that, of course, the great apes are our closest relatives. So if we want to understand the evolution of the parasites that we currently have, the evolution of parasites of, let's say, infectious diseases of humankind, it's really good to understand also how the situation is in our, I cannot say ancestors because these are not really our ancestors, but in our relatives. Just the time of divergence, estimated time of divergence of humans from the branch leading to current chimpanzees and bonobos is roughly 4 million years. So it's a relatively long time, but still these are our closest relatives. So that's good reason. Other reason why to bother is the evolution. Other reason why to bother about the parasites of great apes is also to understand relatively recent changes in the parasitofauna of humans. You probably know that the infectious diseases as we see them today underwent through several significant changes that we call epidemiological transitions. And if you really want to look backwards to the parasites and communities of parasites of our remote ancestors, I mean the early hominids in Africa, there is high probability that these primates were parasitized by communities of parasites which closely resemble parasites of today's savanna chimpanzees. Because it's well known that the lifestyle of early hominids was more or less comparable to the lifestyle of savanna chimps, not to the forest chimpanzees, but to the savanna ones. And surprisingly, the parasitofauna of savanna chimps is quite different. First, from the chimps that are living in the forest, and second, from the parasitofauna of current human population. And the major difference is that basically there are almost no parasites in direct life cycle. Specifically, the soil-transmitted nematodes are almost missing. So it's well possible that great majority of the soil-transmitted nematodes that are currently occurring in the human population are relatively recent invaders connected to the changes of the lifestyle. While savanna chimps have different parasitofauna, it's mainly the environment they are living in and their home range. And this is exactly how our ancestors lived. They roamed in relatively huge open spaces of East African or Central African savannas. Each group has really huge home range, and it's relatively dry and hot environment. So the soil-transmitted nematodes, they really do not have many chances to survive. And the group is instantly moving in a territory which spans like hundreds of square kilometers. And this is why the animals are not reinfected. And this is why only few geohelminths or the parasites with the direct life cycle survive in them. On the contrary, these animals are often parasitized by the parasites that are either vector-borne or involving the insect intermediate hosts, for example, like the spirorid nematodes. So it's quite interesting comparison. And last but not least, of course, the great apes nowadays are endangered or even critically endangered species. They are really iconic species of African and Asian ecosystems. And as such, mainly because of the growing human population, they face really severe pressure or threats. And the major threats for the current or primates is the forest encroachment, logging, hunting for the bushmeat, competition with livestock and agriculture, urbanization. And then there are also two other reasons for growing proximity of great apes, livestock, and the humans, and it's a tourism and the research. And all this kind of underlines the current situation, and basically somewhere there are the major topics or major themes of our research, because what we are interested in is the transmission or exchange of the parasites of great apes, humans, other primates, and even other animals like livestock and so on, to see how much they keep from their original parasitofauna, how much they are acquiring in the areas of close contact with humans, mainly from human population, but also from some other animals. Thanks to all these changes, we see in many areas of the range of great apes, changes in the disease ecology, and of course, it can have negative impact on their populations. When we speak about the great apes, when we speak about the research, when we speak about tourism, or when we speak about contact with humans, we have to talk about primate habituation, because specifically in the great apes, the habituation is really a process that strictly or seriously impacts on their life and on their population. So what's the habituation is? This is kind of course, romantic vision of habituation. Habituation is a process where the wild animals are gradually becoming used to the presence or close proximity of humans, and they are losing principally their original or natural fear of human beings, and they are willing to accept relatively close contact with the human being, by this girl and the squirrels. But as I said, this is a romantic vision of the habituation. But the habituation of the wild fauna and growing proximity of the wild fauna and the humans is occurring worldwide. And we can say that it's really extreme case of coexistence, which is really not usual, and it's something new. Like being in really direct touch, like physical touch with wildlife, it's something that is happening basically just in past decades. It was not happening in the before in such a huge extent. And of course, habituation is important also. It has several important aspects, like it's important for the wildlife conservation, for the communication of the needs of wildlife conservation. Because if we want to teach the people or make the people aware of the needs of the wildlife, we have to show them also the wildlife, of course. So the education, it's one of the major outcomes of the habituation of the wild animals. And of course, the research. Because us as researchers, if we want really to study the alive animals, alive wildlife, we need habituated animals, because otherwise we cannot approach them. And a lot of research, which is nowadays accumulated about the wildlife, mainly the larger wildlife, depends on habituated groups and habituated populations and so on. However, habituation process as such brings a huge range of risks and controversies, and there is ongoing debate. What are the pros and what are the cons of the habituation as such? And specifically in case of mountain gorillas, but not only mountain gorillas, even lowland gorillas and chimpanzees, it's one of the most prominent examples, because in Virunga, in the mountains where we worked recently, almost every group is almost daily visited by a group of tourists. Before the COVID. Yeah. So it's enormous pressure. It's enormous range of daily contacts between two species of primates, between the humans and the gorillas. And of course, it really creates quite intense or dynamic interface for exchange of the diseases. Of course, the first, which are being transmitted this way, are directly transmitted viruses. This is why most of the great ape populations are or were closed for the tourists because of risk of transmission of the COVID-19. But it also creates the encroachment or invasion of humans into the natural ecosystem inhabited or by primates. Of course, brings the risk also of transmission of other diseases. Including the parasitic ones, the protozoan diseases, helminth diseases, and so on. And to be honest, it can happen, of course, in both directions. And I will show you examples that this is happening in both directions, not only from humans to the great apes, but maybe a little bit surprisingly, this is happening also the opposite way, from great apes to humans. So positive, negative aspects of the habituation. Of course, the positive one is the PR, the communication of the needs of the conservation and protection of these animals, education, research, and of course, money. Finally, it's all about money. And of course, the tourism, or you can call it ecotourism or wildlife tourism, which is revolving around the great apes, brings really huge amount of money, which in part, of course, are used back to support the conservation, but they are also important aspect for the local communities and the local governments to understand the need or to understand the importance of wildlife as a source of the income. And the negative aspects of the habituation, first, changes in the ecology and ethology of these animals, because these are not wild animals anymore, to be honest. Because the daily contact with humans, it makes them principally quite different from the really wild living apes. And of course, it's extremely increasing the risk of poaching and other negative aspects of the human population. And it was for me quite a new aspect, or new to understand this, that once we habituate the great apes, principally we have to protect them for the rest of their life, because they are not afraid of humans anymore. And they can hardly distinguish between poacher and the tourists and the researcher. And these animals are the easiest target for the hunters. And this is one of the main reasons, finally, why these animals are under daily supervision of the researchers, of the rangers. Principally, these people are protecting them with their own bodies or with their own presence from the poachers. So this is important aspect to understand. And of course, it's also important to understand kind of responsibility. Once we habituate the group of apes, principally it's habituated, and we must be sure that we have capacities, that we have interest, and that we have money to keep this group habituated and to keep it under protection. And last but not least, those are also the diseases, because of course the habituated animals are much closer to the humans than the wild ones. And they can serve as a kind of bridge between the human population and the true wild population. And as I said, this is true mainly for the viruses, but also the parasitic infection plays some important. This was one of the main targets or aims of our research, both in Central African Republic, Cameroon, Gabon, and so on, and also in Rwanda recently. So when we speak about the diseases transmissible between the apes and the humans, we should speak mainly about the viral and bacterial. There is a lot of information about viral and bacterial infection. A lot of viruses like Ebola, lentiviruses, respiviruses, adenoviruses, coronaviruses recently, and so on are transmissible. And then the phylogenetic proximity that we discussed at the beginning between humans and great apes is one of the major reasons for that. Then, of course, a lot of bacteria are transmissible as well. And but in these cases, it's not necessarily direct. And like in case of anthrax, which I have on the last line, it's not, of course, direct transmission from humans to the great apes. If this is happening, this is usually happening the opposite way, from apes to humans, to the hunters and so on. But in most of the situation, the apes and the humans are facing the same threat from the anthrax being present in the ecosystems. So the question that was intriguing question in the beginning of our research was really, how is it with the parasites? We know about viruses, how is it with the parasites? So within past 15 years, me and my team, we had a chance to work in several field sites with different species of the apes all across Africa, from the West Africa, Guinea-Bissau, Senegal, and so on, the Western chimpanzees. In Central Africa, both the chimpanzees and lowland gorillas. The main place for our research was DSPA, Zangasanga protected areas in Central African Republic. I show you some pictures from there. And very recently, Virunga Mountains and the mountain gorillas, and also mountain gorillas in Uganda and Congo and so on. Okay, so first let's look to the Zangasanga protected areas, which is one of the nicest. To be honest, it's really one of the largest and nicest piece of the wilderness in Africa. It's in Central African Republic, which is country where people normally do not know. And we studied there first mainly malaria, and second the gastrointestinal nematodes. But finally, after 10 years of research, we basically published on all components of the gastrointestinal parasitofauna and blood parasites of these animals. This is Central African Republic, one of the largest countries in tropical Africa and one of the least known countries in tropical Africa, and definitely is the least developed country in tropical Africa. And majority of people even have no idea that Central Republic or Republic Central African exists. The area I'm speaking about is in the extreme southwest corner, this kind of appendix or triangle in the low or bottom left side of the slide between Cameroon, Congo, and Central African Republic. This is the area where so-called tri-national complex of national parks is existing, making one of the largest or probably the largest piece of the lowland Congo forest protected for the wildlife between Cameroon, Central African Republic, and the Congo. Right here. This is how the area looked like from the plane before the landing to the area. So it's really lowland, it's flat area covered by thousands and thousands kilometers of pristine tropical Congo Basin forest, inhabited by a lot of really iconic species. Like it's a home of hundreds of lowland gorillas, but in the same time, it's the most important area of the occurrence of forest elephants. I'm sure you know that nowadays the African elephants are divided to two well-separated species, the large savanna one, mainly in East and South Africa, and then in the Congo forest, mainly nowadays the forest elephant, the smaller one. And it's the first place in the world where the lowland gorillas were habituated. Because the process of the habituation is more complicated and longer than in the chimpanzees. So chimpanzees were habituated ages before the lowland gorillas, and the same is true for mountain gorillas. Like Diane Fossey, she habituated the first groups of mountain gorillas much, much earlier than the lowland gorillas were habituated. And the first place where this happened was this part of Central African Republic. Yeah. As I said, this is one of the major sites also for the research on the forest elephants. This is probably the most famous place to spot forest elephants and to study forest elephants, so-called Zangabai in the national park, where in the same time you can meet easily 100 of forest elephants. On the other hand, the forest elephants are one of the major obstacles for the research on lowland gorillas in the area, because of course, following the groups of lowland gorillas for any kind of research, including the parasitology, you are entering the territories of the forest elephants, and they are not really nice pets. And in many situations, it creates kind of real complication and relatively tense situations between humans and the elephants. Oh, this is just for you to show. As I promised, I show you some pictures from Africa. This is how the field camp looks like. One of the nicest research camps I had a chance to visit, directly in the middle of Zangasanga protected areas, maybe 2 hours driving from the nearest village. This is the spot where the researchers live, and this is the place where our team was working for a couple of years. This library and central lab, kitchen, shower. That was really nice life in there, to be honest. And this is what we were looking for. This is a gorilla poop. Because of course, if you want to do any kind of research on the mainly pathogens or microbiota of great apes, in great majority of cases, you depend on the non-invasive techniques, because it's absolutely impossible to catch these animals. It's really difficult, complicated, and dangerous to immobilize these animals. So we are trying to get as many data as possible from the materials like saliva, urine, and the feces, which you can, specifically from the habituated groups, you can collect using non-invasive methods. And thanks mainly to the molecular tools, but not only the molecular, also the biochemical tools and so on, we are nowadays able to monitor the stress hormones, but we are also able to monitor, I'll speak about it later, such principally surprising pathogens like malaria in the animal feces. So most of the research on malaria of great apes, chimpanzees, gorillas, orangutans depends on the little tiny amounts of the DNA of Plasmodium obtained from the fecal samples, because simply we cannot sample the blood from these animals. And this is the typical situation when the research group follows the habituated gorilla. So this is roughly the distance for which you can approach the gorillas when they are habituated. So there is the rule of the minimum distance, which is usually in different areas. It's a little bit different, but it's something between 7 and 10 meters. So there is not really direct contact. The gorillas, they remain wild and they remain dangerous, like the silverback in this case. This is Macumba again, I think. And basically every day, the research team composed either of the local or the foreign researchers is following the gorillas. Or if not the researchers, then at least the rangers, which of course you can imagine how complicated is the logistics and how expensive is the maintenance of the habituation program and contact with the habituated groups. And this is also why, to be honest, the tourism, the gorilla tourism is so expensive, because it's bloody expensive, all this. Imagine that like in Central African Republic, in this spot, there are 4 groups of habituated gorillas. For every group, you need 2 groups of people following them every day. So every day you need 8 field teams that should be paid for 365 days a year for following these gorillas, supervising these gorillas, keeping the notes, collecting the samples, and so on. So finally, it costs millions of dollars. And of course, the tourism is greatly contributing to that. Okay, so this is kind of distance in which you can approach. This is the shortest distance you can approach the lowland gorillas when they are habituated. On the other hand, if you wish to work with non-habituated ones, even to see them for a couple of seconds in the forest, it's extremely rare situation. And this is why research on the wild gorillas is so complicated. And of course, you cannot identify them. The major advantage for the research of habituated groups is that these animals are distinguishable at the individual level. So you can keep sampling the particular individuals in the long period of time or short period of time, so you know with what is the source, or which is the source of your material you are examining, and then you can try to understand the ecology of the disease. Nothing like that is possible on really wild apes, because you follow the group one day, other day you follow the other group, you cannot distinguish them, you are just opportunistically taking the samples, you do not know to which individual, which sample belong, and so on and so on. So most of the really valuable results on parasitofauna are derived from the habituated groups. So as I mentioned before, first disease we studied or we wanted to study was malaria. That kind of research was inspired by a boom of studies on malaria on apes connected to the discovery of our ability to amplify the DNA of Plasmodium from the fecal samples. This is what I was discussing just a couple of minutes ago. And based on this, we of course wanted to contribute also on our lowland gorillas in the Central African Republic to understand what's the diversity of Plasmodium in the group or groups, what's the ecology of the disease, and if there is any clinical impact, which finally was impossible to study because we still do not have enough data from the even habituated gorillas to really judge or evaluate the impact of the infection. And of course, our other idea was also, or our other aim was to study if there is any transmission from people to gorillas or vice versa. So this is, as I mentioned before, all this is about the fecal samples. So this is basically the result of the sampling trip. The fecal samples preserved in ethanol for DNA isolation, RNA later, also for DNA isolation or for RNA isolation, and in the formalin for the routine coproscopy. And the procedure was principally always the same. You collect the sample in the field, you preserve it directly in the field. Then in the European, you export this load of paperwork. In European lab, you do the DNA isolation, or you can do it locally. You do PCR, of course, cloning, sequencing, finally phylogenetics. So it looks easy, but finally it's a little bit tricky, specifically in case of malaria and the fecal samples. Yeah. So our idea first was to study the diversity within the communities of the primates that are living in the area, but also to study the transmission and also to see, later. So this is the sampling of the local population. So we got the data from the trackers, from the Bayaka hunters, simply the local population living in the forest, including the researchers. We isolated the DNA from the fecal samples of gorillas. We collected the mosquitoes to be able, or to try at least, again, unsuccessfully, to be honest, to identify what are the vectors of the Plasmodium species that are occurring locally. This is one of the traps for the mosquitoes because we wanted to collect specifically the mosquitoes which are being attracted to the primates. So we were not interested in all kind of mosquito in the forest. The diversity is really huge. So we constructed this kind of relatively sophisticated trap, and we used our own bodies as a bait for the mosquitoes. So there was always a volunteer, one of us volunteering, sleeping inside a tent in the forest, and the mosquitoes were attracted to his smell. You can imagine how researchers are smelling after 1 month in the forest. And then we collected the mosquitoes from this outer tent when they were relaxing or simply sitting. This is one of the Anopheles species collected this way. So, and here are the results. Not surprisingly, or at least partly, no, back. We found 3 gorilla genotypes that were already known in the past to be present in gorillas. Here they are named G1, G2, G3. In fact, they can be correlated to the names which are on the right side of the picture. Those are the gorilla specific. Then interestingly, we found also 2 further species, one closely related to Plasmodium vivax, or maybe even Plasmodium vivax, it's still not clear yet. And the second, surprisingly, was Plasmodium ovale, the agent of human malaria. Yeah. We didn't find Plasmodium falciparum in gorillas. And on the contrary, as you see down there, in human population, we found Plasmodium falciparum and identical Plasmodium ovale as we found in gorillas, which creates a first record of ovale in lowland gorillas, making at least suggestion or suspicion of the transmission between humans and the gorillas. So what we know, there is Plasmodium ovale circulating somehow in some extent in the forest among the gorillas. In the same time, we know that the same genotype of Plasmodium is circulating in the human population among the local hunters, together with falciparum. However, it doesn't necessarily mean that there is intensive exchange of these pathogens, but with high probability there is. And in this situation, it's quite probable that those, I guess, 2 or 3 gorillas found positive for Plasmodium ovale were, in fact, infected by the strain of the human origin. So this is at least highly suggestive for the transmission from humans to gorillas. On the other hand, so far as I know, there is no clear evidence of the transmission of primate malaria to the humans. However, what we know, and it was somehow obvious also from this tree, the Plasmodium species which are occurring in the primates from the Laverania group are closely related to Plasmodium falciparum. So after this research, I don't mean research of us, but after accumulated knowledge on phylogeny of Plasmodium in the past, let's say, decade or 15 years, we are sure that the human malaria evolved through the process of coevolution, maybe some host switches also, together with the malaria of African great apes. Okay, so these are the gorillas. And having the team and having the methodology to study the malaria in apes, we moved our research to also in parallel to other locality, which was nice forest reserve protected areas in Uganda, Kalinzo Forest Reserve, where we did research together with Japanese partners. And principally similar to the situation in lowland gorillas, we found also 3 genotypes. However, the different genotypes, these are the chimpanzee specific genotypes of Plasmodium, which are nowadays called Plasmodium gaboni, Plasmodium billcollinsi, and Plasmodium reichenovi. We didn't find any evidence of the transmission, even though there is also human population living around. We didn't find any evidence of the transmission between the chimpanzees and the gorillas. So all Plasmodium we found in chimps was chimpanzee malaria. So speaking about the malaria, the answer to the original question in the title of my talk, how much do we share with the great apes? So in case of malaria, we do not share much. There are probably accidental transmissions of Plasmodium between the apes and the humans in Africa. However, the host specificity is quite strict, and great apes keep their own diversity, which is even higher than the diversity, the diversity of Plasmodia, which is even higher than the diversity of Plasmodium in the human population. What was interesting, having the phylogenetic data, which are accumulated quite a lot, we were able also to see some intra-specific variability, and both in case of Plasmodium gaboni and in case of Plasmodium reichenovi, as you see on the left side of the picture, we see clear split into two lineages, which follows the split of the subspecies of the chimpanzees. Here on the right side, you see the you see the subspecies of the chimpanzees, which divides into four subspecies and one extra species, which is the bonobo. But the chimpanzee as such divides to West African in the Senegal and these areas. Then Elioti, which is in Nigeria, then Central African chimpanzee, which is the nominotypic one, Troglodytes troglodytes, and the East African one, Troglodytes schweifurti. And the lineages that you see here for Gaboni and Reichenovi, they clearly separate the strains or the genotypes or isolates or haplotypes of Plasmodium from Reichenovi, which is the one with the green and blue, the lines with the branches with green sequences of green codes and the blue codes, and the Central African Troglodytes troglodytes, which is the nearest, which nicely supports or shows the coevolution, the long-term coevolution of Plasmodia together with their host, in this case chimpanzee, without host switches and without intermixing with the human population. So these are well separated species. I mean, Gaboni, Reichenovi, and so on, that really their history goes back to the early evolution probably of the chimpanzees as such, because the coevolution with the host, it's obvious from this data, or at least highly suggestive from this data. Fine, so that was the malaria. Years ago, finally, it's almost 10 years ago, we published some of these, the first data, and like 5 years, we published the last study on that. However, what we found more complicated and more interesting are the hookworms, even though they look like, okay, hookworms, the soil transmitting nematodes, nothing interesting. The opposite is, the truth is opposite. This is really interesting group of the helminths. Why? Why particularly strongylids? Why to speak about the hookworms? When we speak about the strongylids as such, principally, this is the group of parasites which is present almost in all mammalian hosts. And what's interesting, specifically in the herbivores, they tend to form the complex communities. The domestic horses can be the nicest example. Look at the strongyles of the domestic horse. There are tens of the species in many genera living in the communities that we basically do not understand at all. We just see the diversity. We know that they live together in some situations, in some maybe not even, but we don't understand the ecology of these communities. Because of the mixture of the species, the diagnostics is usually a real issue. Even when you have the adults, the adult worms, it's complicated. But if you have only the larvae or the eggs, it's almost impossible. And if you look to the primates specifically, it's one of the least studied primate group of helminths, even though it infects almost 1 billion people. If we speak about the human population, because the humans are nothing more, nothing less than other primates as well. Yeah. And at least the species in the humans, they belong to the group of so-called neglected tropical diseases. And what's important, some of them are zoonotic, we know it well. If you look to ankylostoma, Ceylanicum, transmissible between the dogs and the humans and so on. Yeah, so some are simply well shared between the animals and the humans. So what was our aim? Our aim was from the very beginning to use the samples collected in Central African Republic, to use extensive set of samples. Nowadays, we are speaking about the thousands of fecal samples we have from apes all across Africa, and to investigate the diversity of the communities of the strongyles. However, because of expected and proven mixed infections, the standard tools for understanding the diversity are principally not applicable. And the aim of my team in that time, led by, even recently led by Bara Pavcho, my colleague, the aim was to develop the high throughput sequencing, the next generation based approach to evaluate the diversity of these groups of the helminths. So again, in the beginning, the same material like before, the poop, the fecal samples collected, the fresh fecal samples collected in the forest by the field teams. Preservation, in many aspects, in many situations, those were the same samples we used for the studies of malaria. Larvoscopy, that was quite an interesting period. This is so-called Harada Mori cultivation method. Then basic evaluation in the field, advanced microscopy in European labs, finally the evaluation of the morphology and the sequences in the Japanese labs together with Professor Hideo Hasegawa, kind of helminthological guru or mentor of our team from Oita University. So if you look to the situation in general, to our ideas in the general, it goes back to the habituation process, to be honest. Our idea from the very beginning was to study the habituated groups of gorillas, under-habituation groups, and unhabituated groups, and compare with their parasitofauna with the humans. But if you use only the microscopy, basically you are getting the results which are on the right side of the picture. You see the nice spectrum of the parasites. Of course, you see the strongylines, you see their eggs, you see their larvae, but to get any insight about the transmission is simply impossible because you cannot identify them. So the presence of the same worms, or sorry, the presence of the same eggs or larvae in the humans and in the gorilla doesn't necessarily mean that this is cross-transmission and that they are sharing this parasite, because we cannot identify the parasite to the species level. And if we had even more host species, like in this case, those were the manga bees, which are the other dominant primate in Zangasanga protected areas, it's even more complicated. And again, we see the strongyles, like here on the picture 4, which are morphologically identical to those in gorillas, even identical to those in humans. But to understand if they are the same or not, it's impossible without molecular data. And this is showing just the prevalences. If you look to the upper table, the prevalence in humans, almost 50%. The prevalence in gorillas, almost 100%. But the question remains the same. Is there any transmission between them or not? Impossible to say based on the egg morphology. Or even based on the larvae morphology. On the larval morphology, we can barely identify the genus. But again, it principally says nothing about the cross-transmission. So at the beginning of our research, we started with traditional Sanger sequences of ITS and COX-1, isolated from single larvas. That's possible, but imagine the amount of the work. You have to pick the individual larvae, you have to isolate the DNA from them, you have to sequence them and so on. Then the question is how many larvae to pick from one sample if I want to describe the full spectrum and so on and so on. But anyway, even this data brought from the very beginning very interesting data, because we proven clearly in our groups of gorillas that there is Necator americanus, the dominant hookworm of the human population in the tropics, suggest both in the humans and gorillas, suggesting the transmission of the infection from humans to gorillas as a result of the living in the same environment. But interestingly, we found other haplotypes of Necator, which were tentatively determined in that time as Necator gorillae, and we found them to be transmitted the opposite way. Or we found them on both sides, but it was quite suggestive that they are transmitted from great apes to the humans, because they were dominantly present in the human population that is directly entering the habitats of the great apes, like the researchers, interestingly, and the local hunters. The origin of this second species was unclear. So in conclusion, we can say that in the shared environment, there are two types of the Necator shared between the primates, including humans. One on the direction from apes to humans, the other from so it's fair exchange, fair trade, the other from humans to the apes. Of course, in case of Necator, the transmission through the commonly used environment is facilitated by the fact that these nematodes, as you surely know, the larvae are migrating through the skin to the host. So the walking barefoot in the forest or in the sandals, it's enough to get the infection from gorillas, which was nicely proven by most of our researchers, by most of the people in my team, because after being several weeks with gorilla, after living several weeks in the humid environment of the forest inhabited by gorillas, you can be almost sure that you will acquire the infection, which was the background for our second study, when we were able, after the helminthization of my colleague Barbara, we were able to pick the worms from her stool and finally identify the species based on the morphology that was previously believed to be Necator gorilla. So we really proven by morphology that it is really Necator gorilla. So that was the background. But still, we were interested also in the others. What are the other worms that we see? What are there any other? What are the worms of the manga bees, and so on and so on? And then there was impossible. To use it through to investigate that through typical or traditional or old-fashioned, we can say nowadays, Sanger sequencing, because you know that applying the Sanger, there are several advantages and disadvantages. Advantages are well-established protocols. It's very easy for the adult nematodes. If you have a worm from the necropsy or from the fecal sample to sequence COX or ITS nowadays, it's a piece of cake. You do it in 24 hours, and in 3 days, you have the sequence back. But the other advantage is that from a single worm, you have the opportunity to simultaneously sequence several markers. You can do COX, you can do ATNS, you can do whatever, you can do ITS. All this data you can accumulate for one particular individual of the worm. So for now, all good. However, this is a really problematic approach for larvae and individuals because the amount of the DNA is quite low. And second, this is principally unsuitable for the DNA mixture of closely related species, genotypes, or haplotypes. Why? Well, because the primers, they amplify all, and you see the mixed chromatograms, and you cannot basically say, or you cannot, without complicated cloning, you cannot identify the individuals genotypes involved. And this is the space for the metabarcoding using the 18S or other molecular markers through the next generation sequencing. As you know, for sure, the methods was primarily or most or progressively developed for barcoding of the communities of the bacteria. This is the method that serves for obtaining those complex data about the microbiome, which is nowadays a very fancy area of the research. But the same method, of course, it's usable for any mixtures of the DNA if you have universal primers that amplify all. And this was the method that we use for the amplification mainly of the ITS, the internal spacers of the nematodes. What are the advantages, disadvantages? You can kind of dissect the complex communities this way. So first time in the history, nowadays we are using the same for the horses and cattle, and it's even more interesting than the apes. First time in the history, we have a tool how to get real insight into the diversity of these parasites. And you can even pick the rare taxa because you don't see them when you are doing the individual larvae. You cannot sequence 1,000 larvae from a horse or gorilla. So basically you are, if you do the one by one method, you only pick the common ones or mostly the common ones. But with this method, you can get even the rare ones. The disadvantages are also several. Amplicon length restriction, usually you amplify few hundreds of the base pairs of the studied marker and complicated preparation of the DNA libraries and so on and so on. Yeah. So I don't go to the details. This is technical. Let's look to the results. This is how the data are. On the right side of the picture, you see the diversity of the, let's call it haplotypes. In upper picture, it's Necator. Down there, it's Esophagostomum. And on the right side of the picture, you see the heat map. You see the frequency with which the particle haplotype is detected in given species. And even from this was the first round. This was our first trial. And even from this, you see that there are really huge differences. If you look to the Esophagostomum, clearly there is different Esophagostomum or dominant haplotype of Esophagostomum in gorillas, and the manga bees is totally different. This was the pilot. We used only gorillas and the manga bees. Yeah, so apparently there is a species like with high probability Esophagostomum stephanostomum in gorillas and Esophagostomum whatever, bifurcum, I guess, yes, in the manga bees. In case of Necator, it's definitely more complicated. There are apparently some haplotypes which are shared, like for example, the haplotype 1 on the top. Some other are kind of host specific, like the haplotypes on the bottom part of the picture. So that was our pilot that kind of proven the concept and proven the idea, yes, this works. And nowadays we have a range of more. So after that, after having the data from gorillas and the manga bees, we added also chimpanzees and the humans. getting already quite complicated picture. This is one of the first outcomes. Nowadays, we have several analyses like that. And again, you see, first of all, that there's really a range of the genera, which are listed on the right side, and you can divide them, distinguish them by the colors. We found Ankylostoma, Libiostrongylus, Necator, Esophagostome, Ternidens, Trichostrom, other Trichostrongylus, and so on. And again, you see significant trends in the host specificity. Yeah, I don't know if you can read it, but this is Necator, apparently shared between the species. Yeah. If you look to Esophagostomum, this is specific already and not in humans. If we look, so there are different species in the gorillas and chimps with high level of transmissions, but different species in the manga bees. And beside that, we found some that were overlooked in the traditional methods, like Ankylostoma, specific for the humans, and some other which were really rare before, like Trichostrongylus, Libiostrongylus, and so on and so on. So, and that was published already again years ago. So it was kind of first insight into the diversity. We continue the research until nowadays, preparing a new projects on the topic. So if we conclude, there are two ways of the transmission, but what's still neglected and what we really do not know, if Necator gorilla is further shared in the human population. So far, we know that it's present in the hunters or in the researchers, but we don't have enough data from the villages to prove if the hunters serve as a bridge host to bring the infection from the forest to the villages and to spread it further, or if the infection remains restricted to the people that are really entering the habitat of the gorillas. So this is one of the themes for the research that we are still involved in. The same way, having the next generation sequencing approach ready, we just switched the genes, switched the topic, and basically we apply the same methods for the distinguishing of amoebas. Again, trying to answer the question, how is this in Amoeba histolytica in the chimpanzees and humans? It was in Cameroon, this study. Because based on the cysts, again, we cannot really distinguish what the histolytica is, and we cannot really distinguish the diversity of other amoebas. Yeah, so we moved to Ja Forest Reserve in Cameroon. And this is, again, the picture of the amoebas published years ago by my PhD student, Clara. Yeah? And you see that what was originally visible as simply cysts of entamoebas was finally nothing less than almost, I don't remember to be honest, 40 different haplotypes, again with some level of host specificity, with some of them related to Entamoeba suis that were clearly host specific for the gorillas, some other they were more common in the human population, and so on and so on. So it's just another proof that the idea or the NGS approach can really bring us the detailed data. It doesn't necessarily mean that we really understand what these results are showing us. And if this is really what the things that look similar are really similar or identical and so on. So it really needs a lot of extra work. But as a first data, that was interesting proof or insight into the diversity of entamoebas in these hosts. And proving also the common co-infections that you cannot really detect by the routine PCRs and the Sanger sequencing, because you see there were up to 7 haplotypes co-occurring in some hosts and so on. So these are not necessarily details, but co-infections were common. And in the last part of my lecture, I'll take you to the Virungas, the habitat of mountain gorillas, where the research question of our team was a little bit different. I already spoke about it a little in my previous talk that you might remember about the impact of the population densities of gorillas on the spectrum of the helminths and so on. So let's again start with the introduction and some nice pictures from Africa. Mountain gorillas, one of the most endangered primate species, probably one of the most famous primate species, living in relatively small two mountain ranges at the border between Congo, Uganda, and Rwanda. And these two mountain ranges are Virunga Mountains, the larger one, and windy impenetrable forest in Uganda, the smaller one. These animals were considered until very recently critically endangered because their numbers were below 1,000. However, based on the IUCN classification, thanks to their conservation, their status was decreased to the level endangered, which is really a huge success. Because the last census of mountain gorillas that was finished, I think 2 years ago, clearly proven that already there are more than 1,000 individuals. But as we already discussed in my previous talk, that's nice. The population is growing. And I'll show you pictures later. One of the real reasons for that is the veterinary care. We can ask if this is good or not, that these gorillas cannot basically survive without the intense veterinary management. But what's obvious that their population is growing. And in the same time, as I'll demonstrate in the pictures soon, the habitat is not growing. So apparently the population density of these animals is significantly growing without opportunities to extend their distribution range. These are the old well-known pictures. This is Diane Fossey. You probably know all the story of not Sigourney Weaver, but Diane Fossey from the movie Gorillas in the Mist. And what's really interesting in the time when we started to work in Virunga 3 years ago, maybe 4 years ago, one or two of the females that were originally habituated by Diane Fossey were still living in Virungas, because these are really long living animals. So anyway, to study or to understand the diversity and the pathology of these infections, we were invited to establish and to manage relatively extensive network of several research institutions around the world, with the Veterinary University in Brno being in the center and my team. We collaborated with the teams in Australia, United States, Japan, Africa, and so on. And we collected a lot of material, to be honest, together with the field teams. Thanks to the really established sampling effort, we collected, I guess, more than 4,000 samples, almost 4,000 fecal samples that are still being processed. And we are slowly accumulating the data. Again, first time in the history, to be honest, with involvement of molecular tools. What is the diversity? But not only the diversity, but what's kind of also qualitative aspect of this diversity. I'll explain it later. And the major partners for us in this are Gorilla Doctors, a project or NGO established with partnership of UC Davis in California, one of the, as you know, best veterinary universities in the world. These are just a few pictures from the work of the field team that we normally do not take part on. This is the solving of the critical emergencies by the American and Rwandan or Congolese veterinarians in the team. Part of the project is also an orphanage for the mountain gorillas where few individuals are living, but this is not really important part of the project. And of course, they do a lot of rescue situations. They do a lot of transportations of either dead or sick individuals and care and so on and so on. So that is really intensive management, which is good. And of course, the necropsies to analyze the reasons for the mortality. And basically, we got for our research two types of the samples, the worms from the necropsies and the extensive set of the samples that was collected during the 3 years of the project in the field. Those are those thousands of fecal samples. Even though this is Africa, this is Rwanda, and specifically the center where the mountain gorilla project is happening is well-established laboratory facility with kind of, as you see here, relatively fancy techniques, microscope, histology facilities, and so on and so on. So this is really the place to get the data about the parasites. This is how the ecosystem look like. If you are in Rwanda traveling as a tourist, you feel that this is a really nice country. It's green, it's perfect, it's lush green, nice, simply green. But if you understand, to be honest, how the indigenous African habitat look like, you realize that this is really brutally destroyed African habitat because there is nothing native. If you take one tree after another, all these are exotic, planted, or invasive. The same is true even for the grass and so on. So this is one of the most anthropogenic habitats in Central Africa and also or in Africa, in tropical Africa. And also Rwanda is the most populated country in Africa. Again, this looks nice. But even if you go to those little islands, you see first they are deforested. And second, if you are there, you see only exotic plants. You see invasive plants, invasive grass, exotic crops, and so on and so on. So nothing natural. Few remaining wildlife species like the cranes, which are amazing, of course. Thanks to the intense conservation and rehabilitation program, the population is coming back. This is another nice place in Rwanda, just to show this is Gishwati Forest, where the chimpanzee group is surviving. This is it. This is the forest. All around, it's agriculture. So this is basically 2 times 2 kilometers square of the forest, where is a group of chimps totally locked by the human activities all around. And still this is considered nice and protected piece of the land, but nothing more, nothing like you cross it in half an hour by walking from one side to the other. And these are the last remnants of the wilderness in Rwanda. It's just to show that sometimes our romantic visions of or imaginations of Africa are really outdated. Yeah, Africa is highly populated and of course highly impacted. The same way like Europe, it's not wilder than Italy or not wilder than the Czech Republic. And this is piece of the Virungas already is Mohabura, my favorite mountain, the most impressive mountain from the chain of Virunga mountains. And again, if you look at that like this, you would say, wow, such a nice tropical mountain. But if you look at this picture just taken in the evening where there were no clouds, what you see is nicely the the margin of the national park, because the line that goes between the deforested and the forested, it's very sharp. This is the fence. Basically, this is a wall. This is a stone wall. On one side, it's highly inhabited or densely inhabited agricultural land. Inside is the natural park where the gorillas are living. But the gorillas are not going that high. They can live or they can reach even the summits of these peaks, but there is no habitat for them. So basically what gorillas have, they have only a little tiny ring of the forest, a mountain forest, that goes all around the mountain. So the habitat is really small. I'll show you another picture. Here it is. Yeah, this is the same. The Mohabura was somewhere here in the northeast. So these are the Virungas, and this is the density of the population of the gorillas. So you see that the gorillas are living all around, very close to the border of the national park, where still the forest is located. By the upper part, in this case, I mean the high elevation part, in this case, the central part of this orange spot, and they are only time to time invading that or time to time visiting that. They can climb the mountains, of course, but it's cold and there is really not food, and they are not that much interested in nice view and so on. So they don't go up. So you see that it's really restricted little tiny habitat all around. And if you imagine that the population triplicate in this little ring of the forest without any ability to extend the range, then it's obvious that the density is growing. These are just, again, the pictures from around. People commonly ask me, okay, and cannot the park be extended? And the answer is no, probably cannot, because these are the farmlands. Just for you, interesting, maybe as for parasitologists or veterinarians, the plant here, which looks nice, is pyrethrum. It's the original, it's Compositaceae plant, from which the pyrethrum as a insecticide was originally derived and is still being produced. And this is not the only crop around. There are the potatoes, there are the other crops around. So it's really intense agricultural environment. We did, of course, a lot of public awareness trainings of the local staff, establishing really the coproscopy lab. We use the goats to train the people in the collection of the worms in the necropsies, because the necropsies are not that common of the gorillas, of course. So that was much easier to use the livestock for the training and so on. Is Professor Lapeta from Sydney and my colleagues. This is just to show how the field work is. These are the really high mountains with a lot of clouds, a lot of mists. And this is the environment in which the gorillas are living. So it's not really nice environment, to be honest, like temperature-wise. There is 10 degrees, wet, humid, windy, and so on. Okay, so this is just the Afro-Alpine zone, which is visited by gorillas regularly, but the gorillas are not living in. This is like already, let's say, 2,000 meters. And this is the view down, like really confirming how destroyed the landscape all around is. So if you are gorilla looking down, you obviously see, okay, there is no space. There is not any more space for me to live outside the park, and this is true. These are just a few pictures of by my colleague taken. And this is another interesting picture. It's a Cercopithecus, so-called golden monkey, that is living syntopically. Is the only other primate living syntopically with mountain gorillas in the forest. Quite nice, impressive, endangered small monkey species. These are just a few pictures from our work. Necropsies, the anoplocephalic tapeworms, strongylids in the fecal samples. And of course, one of the major answers behind our research is how serious the changes that we see are. Or shall we react somehow? Is it already time to start deworming the gorillas and so on, but I'm not going to answer them. And because all these parasites, they have some environmentally resistant stages like the larvae or the stages cysticercoids and mites in case of tapeworms, they can be considered soil transmitted, and as such, they are density dependent. And it was the major topic to investigate. We did a lot of complicated statistics, first creating the methodology, trying to assess the homogeneity of the distribution of the eggs in the fecal samples and so on. So that was first year. And sooner or later, we developed a relatively robust methodology that enables us to first investigate the egg counts, which means the intensities of the infections, or to use the numbers of the eggs as a proxy for evaluation of the infection intensities, using the mini flow tag system developed in Italy. You can be proud of that. Of course, the traditional microscopy for the egg identification. These are the first results that were really surprising even for us, showing how different are the egg counts between the gorillas living in the low population density area, which is the blue one, and in the area of high population density. So these are really, and you see that some of the animals are reaching really high level of the eggs. 6,000 eggs per gram, this is already really significant. If you compare it to the livestock, yeah, the 6,000, 7,000 eggs, this is already super infection almost. Yeah. And then we started to do the molecular work and the morphological work on the worms from the necropsies, which is definitely not finished. We've proven several species and even genera of the nematodes living in there. Originating from two major groups. There were the trichostrongylids, which was Heostrongylus and Trichostrongylis, and Murshidea, which is totally different species of the nematode, typical for African herbivores like for the elephants. Yeah. What was interesting that one of the Trichostrongylids was identified as Heostrongylus rubidus, which you know is a porcine parasite. And now we are trying, with involvement of the samples from the domestic pigs, we are also trying to answer the question, okay, how is it? Is there any transmission between the domestic pigs and the gorillas? Not answered yet, to be honest. Yeah, so this is still ongoing research. And this is already interesting, and this is the most interesting, and this is what I want to show you. This again, the next-gen data, the relative abundance of the individual sequences. So longer the column is, more DNA was, more amplicons were detected in the given sample. Individual groups of gorillas, and by the colors you can see the different types of the nematodes involved. And what's really interesting is that in different part of the range, the composition of the fauna of the strongylids is quite different. If you look to the central part, to the groups from Rwanda, which are the groups like Agasha, I cannot read it, Sabino, Muheza, Hirwa, and so on, you see this absolute dominance of the blue color, which is Murshidea, which is non-pathogenic strongylid. However, if you go to the more to the left, like to other groups, which again, I cannot read, but Kureba and Susa and these groups, you see the dominance of the red color, which is the Heostrongylus, which we know is having the larvae deep in the tissues, causing mainly gastritis or intestinal inflammations. So this is something that can be really pathogenic. And if you look to the absolute right side of the picture, you see a lot of the black color. This is already the second subpopulation. This is the mountain range in windy impenetrable forest. You see a lot of black, and what's black? It's esophagostomum, the parasite which is able to form the cysts in the intestinal mucosa. Yeah. So what this means is difficult to answer. We are gradually publishing the results, cumulating the analysis and so on. But if we look to this from the two angles of view, the first is traditional and the second is the advanced approach. The traditional approach will show us all gorillas, they have high loads of EPGs. So basically the gorillas from the groups in the central part of the picture and gorillas on the right part of the picture and the left part, they can look the same under the microscope. They have high EPG loads. But what's important, and we don't know why, some of the gorillas, they have high loads of some groups, they have high loads of non-pathogenic Murshidia. Some other, they have high loads of pathogenic Heostrongylus, and some other in the north, they have high loads of the esophagostomum. So as for us as veterinarians, this is already showing something, that the picture of the disease, the resulting picture of the disease, doesn't depend that much on the EPG as such. And to be honest, the same is true, of course, for the domestic ruminants. The same is true for the horses. Like having the high EPGs for the small and large strongyres, it means really different clinical consequences. Here, I just want to show you those two subpopulations of mountain gorillas. The one in Windy Impenetrable National Park, which is in the north, and the one in Virungas, which is in the south. The orange spot is the southern part of the range. You see this. Is this the one I spoke about? These are the Virungas. You see the same shape, yeah? The orange and the gray. If you look to the picture here, the groups of gorillas which are blackish here, they all belong to Windy Impenetrable National Park. The remaining groups belong to Uganda. Sorry, to Virunga. And we can ask, okay, so what are the differences between these two subpopulations? Interestingly, originally, they were connected. So the fact that mountain gorillas are nowadays split into the two populations, it's again, result of anthropogenic pressure. And the lowland between these two mountain ranges is already so destroyed and highly populated that the gorillas are not living there probably for many hundreds of years or many centuries. And the other difference is, of course, the type of the ecosystem. And other difference, which probably answer the presence of the esophagostomum in Windy, is the presence of the chimpanzees. Yeah, in Windy Impenetrable Park, there are the chimps living together with the gorillas and the humans, of course. In Virunga, the gorillas are living without the chimps. So for now, the hypothesis, and chimpanzees are quite commonly infected by esophagostomum. This is what we know in general. So the hypothesis for this, or the first hypothesis we have for the, let's say, one of stronger hypotheses we have for these differences is that in windy and impenetrable forests, the gorillas, they share part of the parasitofauna, namely esophagostomum, with the chimpanzees. Question is how much this can impact on the pathogenicity, how much this is involved in so-called chronic wasting syndrome, which they observe in windy. So this is the topic for the next grant or another grant my colleagues are having starting from now. But it's well possible that this is kind of this is an aspect or attribute that should be considered in evaluation again in the pathogenicity of the strongylids or not. Or strongylids, if they are pathogenic or not. That is the same picture again. And maybe we still have a few minutes, so I added some more pictures and some interesting data. The Gorilla Doctors, the NGO we collaborate with, is one of the important partners of the project by UC Davis that just finished, I think, a year ago, which was called PREDICT. I don't know if you heard about it, but PREDICT was, as you see here in the short text, a global surveillance of the pathogens, specifically targeting the wildlife in the tropics. It was probably the largest project in the human history devoted or directed to the detection of the viruses in the wildlife and even in the livestock. And these are quite interesting data. I was, I think, speaking about them even in my last lecture. And now look to the numbers in the lower right corner. Yeah. And for me, this is really something showing where we are as humans with understanding on the diversity of the viruses. Yeah. So the research team as such managed to discover, including the material from virungas and from gorillas and so on, to discover almost 1,200 unique viruses. And out of them, more than 900 were new. So the great majority of the diversity of the viruses in the animals, we are speaking about the mammals. It was not about frogs, reptiles, insects. No, that was about mammals. So still the great diversity or the great part of the diversity of the mammalian viruses is basically unknown. And with all the anthropogenic changes or on all the intense transmission of the pathogens or contact between the humans and the wildlife, of course, the interface is getting more and more intensified or intense, and the chance of transmission of these diseases is increasing, which is perfectly documented by COVID-19, because this is the virus that exactly emerged from this environment. Subtropical environment, close contact between human population and some wildlife, even though we don't know exactly which one. And we have an issue. What's also for me impressive, to be honest, on the PREDICT project is the massive size of this approach. As you see here on this very simplified graphics, they established and equipped more than 60 labs in tropical Africa and Asia, and sampled more than 145,000 of individuals of different mammals. So this is really something. Also involvement of the training, like I mean the virology or One Health training of 6,000 people in 30 countries within one project sponsored by USAID. It's for me something really impressive. And in my opinion, this is something that is really needed if you want to understand the diversity of the pathogens. Doesn't matter if these are viruses or bacteria or even the parasites, but if you really want to understand what's going on, we should really use massive approach, not the little teams like my team, 10 people scattered in the forest somewhere for ages. That's interesting. It brings interesting data, but only if we put it into context of really global research. Maybe a few more pictures at the end. Together with the PREDICT team, we had a chance to do necropsies also of the bats and wild rodents and so on in the same time where we were working in the gorilla. So it was for me really nice. I always love the work with bats. So it was really, really interesting to see the field team sampling the tropical bats for the parasites. And for me, it was also a really nice opportunity to see two unique ectoparasites of the bats. And this I want to show you as a little quiz. So this is a bat of the genus Optomops, one of the typical, very impressive, quite large, strong tropical insectivorous bats. And this is the second dominant species. This is the fruit bat, Rosetus aegyptiacus, the well-known fruit bat species. And this is the first thing I want to ask you. What do you feel this can be? I had no idea in the time when I was taking the picture. So this is on the wing of a fruit bat. No, it's a wing of Optomops or fruit bat, doesn't matter, it's a bat. So do you have any idea what this can be, just to keep kind of contact? I still hope you are there. Any idea? Little cysts, like 4 millimeters, maybe 5, with something inside, which is partly coming out.
I don't know. I confess.
I also didn't know, to be honest. Try someone.
Maybe a butterfly?
Pardon me?
Maybe a butterfly?
That's a good idea. It was also my suggestion, but it is not. Here it is coming out, basically something almost amorphous coming out of the skin if you do the necropsy. Not necropsy, if you do to try to dissect it out. No, you probably didn't, never heard about that, and neither I did before. These are the dipterans. These are the strablid dipterans, parasitic dipterans of the genus Ascodipteron. And what's interesting, these are the wingless females. It's a little bit similar like to Tunga, the biology of that. You know, Tunga penetrans, the flea which is living under the skin. So this is a fly living under the skin. And what's interesting is the history, how it is happening. At the origin, the female has wings. It flies. It finds a bed. And then it burrows herself under the skin, losing the wings, losing the legs, making basically similar to Tunga, very amorphic structure, which is only for the production of the offsprings. So you don't really see the chitinous structures. You don't see the legs. You see nothing, and still it's a female of a dipteran. So it was one evening full of discoveries. In that it was like 2 years ago. So it is the first parasite I never heard about. But then we collected these bugs on one of the beds, which was something again really surprising for me. I thought, okay, wow, discovery, wow, something really new. I was almost shaking collecting that. This is it again. So try to identify, at least to the group. It's an insect apparently. So what do you feel it can be? First of all, the number of the legs is not usual, to be honest. Yeah, you see.
Yeah, 4.
There are 4. So no, it has 6 legs. But the first pair is little tiny, and it's under the belly, or under the thorax. So it has 6 legs. So it's an insect, but only 4 legs are visible.
We are writing in the chat, but please speak, because David can.
Yeah, I can open the chat, but just wait. But don't. Coleoptera, no. Lows, no. Blister beetle, no. It's Heteropteran. Yeah, it's a specific family of Heteroptera, the bugs, which is called Polyctenidae, with several species on the beds. And this is Hippoctenes clarus, the blood-feeding or blood-sucking bug living permanently on the beds. So it's not a Cimex, or it's not Cimicidae. As you see, it's not related to not much related to bedbug, but it is still a Heteropteran. So again, for me, something totally new. I saw it first time, and then I realized that it's known for the centuries almost. I think that Hippoctenes clarus was described in the beginning of 20th century, or maybe at the end of 19th century. So it's really well known. So it just shows the limits of us as parasitologists. And what's even interesting, I checked the literature, and nowadays there are even studies on the microbiome of these Heteropterans. So the humans are really accumulating the enormous knowledge recently. Little tiny Heteropteran on some tropical beds somewhere in tropical Africa, and we already have the data about the microbiome of this insect there. So it's, for me, quite interesting or even impressive. And I think that's it. That's it. This is already the slide you've seen. So this is it for today from my side. So if you have any questions, please, better you ask instead of writing to the chat. Feel free to ask.
Thank you, David. It was so interesting. And I have some question here in Facebook, but as Harold is already in the classroom, he probably can address directly to you by speaking his question.
Tell me.
Harold?
You can read that from the Facebook, or?
Yes, I can, but he is also in the classroom.
Yeah, he is. So pardon me, he is in the classroom. Okay.
Yes,
so
yes.
Feel free to ask.
Anyway, I will read from Facebook. How do you prevent resampling the same animals? So this is regarding sampling, I think, gorillas.
That's perfect question. And in case of habituated ones, it's relatively easy because the samples are collected directly by the visual contact with the animals. So they are the rangers or the research team. They know each individual by the name, and they observe them. They keep watching them all day. And when the animal is defecating, they pick the samples. So this is one part of the samples we have, so-called, we call it identified samples. And then as a parallel approach, we collect also the fecal samples from the nests, the night nests. The gorillas, both the lowland gorillas and the mountain gorillas, they make a nests on the ground commonly or in the lower branches of the trees. And they tend to defecate either in the nest or around. In that situation, we know which group it was, but usually we don't know which individual it was. So these are the samples which are considered at the group level. So these are two ways how to do it. Third way that we applied in one of our studies in Tanzania are the genetic tools. If you are following the same group for a long period of time, you can develop basically barcoding method for the individuals. So once you isolate the DNA from the feces, you can identify which individual it is.
Yeah. Yes, because he already asked you about fingerprints. And he also addressed another question. Do you study, I think, only Strongyloides or any Ascaris Lumbricoides, I think?
Not really, because Ascaris, to be honest, is not present in the wild populations, or it's extremely rare. I saw it few times, and it opens nicely the question of the origin of Ascaris Lumbricoides. And because you know probably that Ascaris Lumbricoides is almost identical genetically to Ascaris suum. And considering that it is absent in the wild apes in Africa, it's highly probably originating, or it's spilled over to human population from the Suids. So it has nothing to do with the great apes, and it's probably not really original human parasite. And this is maybe why it is so pathogenic, because it's difficult to say how long the humans are living with Ascaris Lumbricoides, but probably this is not for millions of years like other worms. So we didn't see Ascaris in the wild populations.
Yes, there is a reply to you in Facebook chat by Serena Cavallero from Roma La Sapienza. He wrote, we have some observation from neotropical primates of Ascaris Lumbricoides, but not from Africa.
Yeah.
It's just a reply to you.
That's interesting, but it can be also a result of the spillover from humans to the primates. Because in the zoos, in the zoological gardens, we know Ascaris in the primates, but specifically in the history. But the hypothesis behind usually is that they acquired the infection from humans, like by direct transmission or through the environment. The same is known from some sanctuaries in Africa. Yeah, so speaking about the wild gorillas, or chimpanzees, I want to stress there is no Ascaris. If you look to the captive ones in Africa, or if you look to those in the sanctuaries, in the orphanages and so on, we know Ascaris in there. So probably those are the humans bringing the infection to them.
Yes, he agreed with you, and thank you for your reply. And I have a last question from Harold from Israel. Mm-hmm. How do you explain high strongyle fecal counts when gorillas are predominantly browsers? And and I have another question. This is from me regarding this very extremely high fecal account. If you ever notice any clinical manifestation of this kind of parasite, because the numbers you have explained for livestock are incredible. Probably they are die.
Okay, yeah, but again, like depends on if we start with the egg counts. Depends on which species. If we see the high egg counts by Murshidea, I don't really think that this is pathogenic. If we see high counts of trichostrongylus, the Heostrongylus and Trichostrongylus, yes, it's responsible for some infections, and there are some cases of gastritis in the necropsies, and there are the cases when gorillas are developing, in Uganda they call it chronic wasting syndrome, and it's empirically known that if you deworm these individuals by macrocyclic lactants like ivermectin or similar, they improve relatively fast. So this is indirect confirmation of the pathogenicity. So yes, it can be pathogenic. And the question of how it is connected to the obtaining the food, I couldn't really distinguish if they are grazers or browsers or whatever, because they take a lot of plants like the ground vegetation, they eat it directly, they pick it, but not by mouth, they pick it by hands, and they commonly eat it even with the remnants of the soil on the roots and so on. Yeah, they eat bamboo, which they eat the young shoots of the bamboo, which are basically just at the ground level. They even take it a little bit maybe from the underground with the roots and so on. So they probably eat a lot of the ground vegetation. And because they are terrestrial, this is the difference from other apes. The mountain gorillas, they almost do not climb the trees. And if they do, it's more for fun than for feeding. But we still don't know the concentration of the larvae in the environment, to be honest. So we really don't know where the larvae of strongylids are, but they should be, in my opinion, everywhere. If you imagine that the gorilla has 7,000 eggs per gram, and the large one can produce 2 kilos of the feces a day, basically it shows millions of larvae being produced every day by these individuals that we can consider super shedders and so on. So probably it's highly contaminated, and it's the optimum environment for the development of larvae because it never gets dry. Even in the dry period of year, there is always some humidity in the soil, so the larvae can survive. But we don't know much about that. We never tried. We had no capacity to collect the larvae in the environment.
Thank you, David. Can you stop sharing your screen? Because this way we can see also picture of people. I have a question from Alireza from Iran. Alireza, you can do it directly.
Hello, thank you.
Hello, Alireza. How are you?
Hi. I have a question. In one of your slides, I saw an oxyurid egg. Was it Enterobius vermicularis or what?
It's a bit more complicated. It was in manga bees, if I remember well, or maybe not, I don't know. But chimpanzees, they have their own. They have Enterobius anthropopitheci, which is a sister species to Enterobius vermicularis. The gorillas, they do not have the pinworms. So to be honest, Enterobius, based on this fact that our human Enterobius has the closest relatives among the chimpanzees, it shows that it's one of our oldest parasites, probably. These are the parasites that are sometimes classified as a heirloom parasites. These are the parasites that we inherited. And it's quite interesting also the way of transmission, because if you imagine that the Enterobius is not soil transmitted nematode, so it can really persist in the nomadic mammalians. And this is how it probably persisted in the nomadic groups of early hominids, because they were permanently moving, but the way of transmission doesn't depend on the soil. So it's the oldest, maybe.
And this is also a question of reverse transmission from humans. But you say that gorillas do not have this infection, or they cannot get infected, or what? Or they were not exposed to?
What I know, there are no data there are no positive findings from gorillas, but there are the data from the zoological gardens about presence of Enterobius vermicularis in the chimpanzees. And in some cases, it was even pathogenic, but these are quite old studies, I think from 60s or 70s, maybe 80s, where there were some individual chimpanzees in the zoological gardens that were considered even dying because of human originating Enterobius vermicularis. So it can happen, but normally in the wild is not happening much, as you said, because of the way of transmission. Normally, there is really not that intensive contact between chimpanzees and humans that enables transmission of Enterobius.
Thank you very much.
Welcome.
Any question from my students in the classroom?
Yes, I have one. First, thank you for your presentation. It was very amazing. And I have 2 questions. The first is, you told us that natural parks are not so big, and you think that the animals that are forced to stay closer between themselves, this can help the spreading of the parasite? And the second is, You didn't mention about an active management of the parasite from human because you think that wildlife is wildlife and they manage the parasite from themselves, or
no,
there are some action that the human can do? And thank you.
These are two questions. I start with the second one. Of course, we can manage this, but question is, it's more philosophical question, really. The question is if we want to do so. Of course, from my perspective, if we really consider wildlife, if we really create a national park to keep the animals protected from any activities of the humans, then ideally we should protect them also from this impacts. Yeah. Even though we are veterinarians, like I would prefer not to do it. But unfortunately, by the continuous pressure to the wildlife, we created the situations in which the animals can survive only under the management, which is like bright and dark future in both, like for veterinarians, like maybe bright for veterinarians because more and more animals will probably require the assistance of the veterinarians. But for the wildlife as such, in my opinion, it's something that we really don't want to see. It's like, I feel kind of sorry because this way we are transforming the gorillas from wild animal to the farmed animal. And honestly, like Virunga, even though people, they don't want to hear that, Virunga is almost a farm for gorillas, which is sad because it's the last place where they can live and still it's not real wilderness. And now I forgot the first question. It was...
The first question is about that the national parks are not so big and the animals to stay closer.
Yes, not only to stay closer, but to contaminate the environment more, because all these parasites we study are soil transmitted, which means this is the contamination. It's not direct contact between the animals, but it's the contamination. It's like in the sheep and horses. If you have a small pasture, if you lock the sheep in the small pasture, the EPGs go high much faster than in the large pasture. Yeah, so this is so-called density-dependent transmission. So you are right, it's about the density, but it's not about the contact as such.
Okay, thank you very much.
Thank you, Davide. Any other question from the classroom? I have a question from my students because probably they are a little bit shy, and if anybody of them would like to Ludovica.
Yes, I am one.
You are very.
Don't you think?
Ludovica, you have to switch off your video because you are very.
I don't understand. Yeah.
Very low connection.
Perfect. Now better.
Okay. Do you hear me now?
Yeah.
Yes.
Perfect.
Okay. I wanted to ask. Don't you think it would be better for tourists to wear gloves or masks or protective clothes to avoid transmitted pathogens?
Yes, this is a huge discussion. And in most of the places in the world nowadays, the tourists, they should wear the masks. And maybe now with the COVID epidemics, it would be probably a mandatory to wear a mask to prevent the transmission, mainly from humans to the animals. The gloves, to be honest, I don't think they can help really much because most of those, if we speak about the viruses, they are respiratory mainly. So the gloves are not helping. Anyway, you are not touching the gorillas. If you are speaking about the parasites, then it's a little bit easier, like to protect the apes, because most of the parasites, they are shedding in the feces. So the basic rule is for the humans, don't defecate in the forest. Yeah, the tourists, they usually don't do it because they stay short. Honestly, if you go for gorillas, you pay usually for 1 hour. So within 1 hour, you commonly don't need to defecate or to poop. But for the rangers, for the soldiers which are in the forest and so on, protecting the animals, making the protection from the humans, managers and so on. So it's a little bit more tricky because these people live really in the forest, and then different national parks, they imply different level of the rules, what you are allowed to do in the forest or what you are not allowed to do. But to be honest, if you are there working for days and days in the forest, you can hardly defecate to a plastic bag. Yeah? So, but there are some rules. So this is it. And of course, if you want to prevent the transmission of strongyles from gorillas to humans, you couldn't wear the sandals. But on the other hand, if you are living in the forest for half a year, you can hardly walk in the rubber boots 12 hours a day for half a year. So sooner or later, you anyway start wearing the sandals or going barefoot in the water and in the camp and so on, and then you catch the strongyles. Yeah. So, but you are right, there are some ways, but the respivirus it's something which is really threatening the apes, and the mask is mandatory, and nobody knows how will the rules develop after the COVID-19.
Yeah, but not only for the COVID-19, but maybe for all the other pathogens.
Yeah, but these are mainly the viruses. Yeah, so don't defecate in the forest is the first rule. No, I don't. We are losing you again. Sorry. But I hope I answered.
Yeah, I think so. Ludovica, we cannot hear you.
No, I'm hearing you.
Okay, good. But we are saying nothing, so I hope that this was enough. But you are right; it should be improved. In many places of Africa, it should be improved. But to be honest, the tourists are not that risky, specifically for the helminths, because they are coming from the really Western societies where helminths are anyway absent. So the probability that some of the Europeans or Americans or whoever from the industrial part of the world brings the worms, it's almost zero. So this is usually the local population. And a lot of national parks, including the Virunga, they have really huge programs for the health monitoring of their employees and also deworming programs for the humans in the villages. Yeah, so there are the ways how to decrease the infection pressure by the human nematodes.
Thank you. Any other question? We have time for doing that. In the meantime, that we think, my question before was if any of my students would like to participate in some weeks, some months of training with your group, do you think it's possible to make an experience like that or not? This is because it was really, really fascinating, and I think that maybe even with COVID, some people would like to join. It's a dream.
It really depends on the situation in the place, because even for us, like working with bats in Rwanda, it was just an accidental participation on the project of the gorilla doctors that was happening there. With gorillas, we are not sure how much we will continue in Virunga, but we are going to continue in Vindi in Uganda. So theoretically, yes, but be ready that it's really complicated, because even for us, unfortunately, getting the research permit and getting the people in is not easy. But theoretically, yes. And for example, we have some small project in Somalia where this can be easier, but not with the apes. So yeah, please try to write me or keep pushing me. I'll try to help you, but don't expect much because it's difficult. It's difficult, and it's also expensive, to be honest, because even though it sounds unbelievable, usually us as researchers, we even pay for doing the research in there. Yeah? It's a common in African parks that you as researcher, you pay daily fee. Yeah, so finally it's becoming really expensive because you pay the flight, you pay all the accommodation, you pay the food, and you still pay the entrance fee for researcher for every day that you are in there. Yeah? So finally, you can easily end up with 5,000 euro for a month, which is relatively expensive.
Yeah. Thank you. Okay. Any other question? Seems no. So, many thanks, David.
Yeah, my pleasure.
Now we wait you here.
Hi, Alireza.
We wait you here in Sardinia.
I look forward, and we can do some research together. I really look forward, and if not now in spring, definitely in late summer or early fall, like September, October, I hope I will come.
Perfect.
Super.
So many thanks again, and have a great day. Thank you.
Ciao, ciao.
Thank you very much.
Thank you very much.
Bye.
Thank you.
Thank you.
Thank you. Bye.
Bye. Bye.